Good day, everyone. Today, we're diving into the critical role of transgenic mice in cancer research. I'm Joyce, and as we explore this topic, we'll uncover how these genetically modified models are revolutionizing our understanding of cancer and driving new therapeutic innovations. Let's start by setting the stage with some key background information. First, let's talk about the global cancer landscape. With populations growing, aging, and risk factors evolving, the cancer burden is rising, making urgent the need for better prevention, diagnosis, and treatment. Traditional therapies like chemo and radiation have limits. They're not always effective, and patient responses vary widely. That's where mice come in. Their genetic similarity to humans, short reproductive cycles, and shared physiology make them ideal models. They bridge the gap between lab science and human relevance, allowing us to test ideas quickly and systematically. Now, what exactly is a transgenic mouse? Simply put, it's a mouse whose genome has been altered by inserting a foreign gene, called a transgene. This gene can come from another species, or be a modified version of the mouse's own genes. The goal? To study specific gene functions or mimic human diseases, especially cancer. By engineering these mice, we can observe how genetic changes lead to disease development, which is crucial for unraveling complex biological processes. Let's explore how we create transgenic mice. 3 common methods. First, pronuclear microinjection, injecting DNA directly into a fertilized egg's nucleus. Second, retroviral transduction, using viruses to deliver genes into embryonic stem cells. Third, CRISPR-Cas9, the revolutionary gene editing tool that lets us precisely modify the genome. Each method has its perks. CRISPR, for example, is highly efficient for targeted edits. Regardless of the method, ensuring the transgene passes to offspring, germline transmission, is key to establishing stable models. Transgenic mice shine in creating cancer models. There are 2 main types. Inducible models, where we control when and where oncogenes are active, think of it as a switch for tumor growth, and spontaneous models, where tumors form naturally due to genetic tweaks. These models mirror human cancer in genetics, pathology, and behavior, letting researchers study disease progression in a controlled setting. They're not just replicas. They're windows into how cancers start and spread in real biological contexts. But their uses don't stop at modeling. Transgenic mice help us dissect gene roles. By adding or modifying genes, we see how they drive cancer initiation or suppression. Equally important is studying the tumor microenvironment, the cells, proteins, and structures around tumors. This environment fuels cancer progression, and these mice let us analyze interactions between cancer cells and their surroundings, like immune cells or blood vessels. Together, these studies reveal molecular and cellular mechanisms that could be targeted for therapy. Crucially, transgenic mice are platforms for testing anti-cancer drugs. We assess efficacy, how well a drug shrinks tumors, slows metastasis, or improves survival, whether it's chemo, targeted therapy, or immunotherapy. We also study toxicity, seeing how drugs affect healthy tissues and metabolism, which is vital for safety. In preclinical development, these models mimic human responses, helping prioritize drugs for clinical trials. They're the bridge that turns lab discoveries into potential treatments. Let's balance the picture. Transgenic mice offer incredible advantages: precise disease modeling, gene study, and drug testing. But they're not perfect. Species differences mean some responses won't mirror humans exactly. Transgene expression can be artificial, and models might not capture all human complexity. Plus, they're costly and time-consuming to develop. Still, their benefits far outweigh the challenges, especially as we refine techniques to address these limitations. Let's look at a real-world example in lung cancer, a leading cancer killer. Researchers used CRISPR to create a mouse model with a KRAS mutation, common in human lung cancer. They tested a targeted therapy and found the mutation drove tumor growth, while the drug significantly slowed tumors and improved survival. This shows how transgenic mice validate genetic drivers of cancer and evaluate precision therapies, bringing us closer to personalized medicine for lung cancer patients. Another case in breast cancer, a heterogeneous disease. Scientists knocked out an immune regulatory gene in mice to study the tumor microenvironment. They found this molecule promoted cancer by shaping immune cell interactions. The insight: targeting immune pathways could boost anti-tumor responses. This highlights how these models uncover microenvironment secrets, paving the way for immunotherapies tailored to breast cancer subtypes. The future of transgenic mouse research is exciting. Advanced CRISPR tools will enable multi-gene editing and epigenetic tweaks, creating more complex models. Humanized mice with functional human immune systems will revolutionize immuno-oncology. We'll see organoid transplants, merging lab-grown tissues with mouse models for realistic tumor studies. Advanced imaging will let us track tumors in real time, and avatar mice, multiple models from one patient, will speed up personalized drug testing. The possibilities are endless as technology and biology converge. At Creative Biolabs, we're proud to support these advancements. Our services span custom model development, from Transgenic to humanized models, to rigorous efficacy testing for CAR-T therapies, oncolytic viruses, and more. We use cutting-edge techniques like multi-omic analysis and imaging to ensure translational relevance. Whether you need a tailored model or comprehensive drug evaluation, we're here to accelerate your research. Visit us at our website or email us to learn more.